can see in this curve. So an increase in the partial pressure of carbon dioxide will shift the oxygen dissociation curve to the right. The partial pressure of carbon dioxide is high due to the tissues actively respiring and to produce ATP for muscle contraction. So at higher partial pressure of carbon dioxide, the percentage saturation of hemoglobin is lower, and hemoglobin has lower affinity for oxygen. So this first question, the figure shows the oxygen saturation curves for hemoglobin under different conditions. Curve A represents the normal resting conditions. Suggest two factors which could be responsible for the shift of the curve from A to B. So the first thing we can talk about is that curve B actually has a higher carbon dioxide concentration, or I can say it has a higher partial pressure of carbon dioxide. And then the other thing you can talk about is because carbon dioxide gas is an acidic gas, you can talk about its acidity. So you can say that it has an increasing acidity, or also, or a decreasing pH. Okay. So for the next question, this is the answer. So for the next question, explain the physiological significance of the shift from A to B on the behavior of hemoglobin in the tissues. So the first thing we can talk about is how that from A to B, the shift is basically caused due to the lower partial pressure of carbon dioxide. So you can say that during respiration, the partial pressure of carbon dioxide rises. So the partial pressure of oxygen gas falls since it's being used in the tissues. So the next one I can talk about is in curve B, the release of oxygen gas from the hemoglobin to the tissues is easier, and the uptake of oxygen gas into the hemoglobin is easier. So that
hang on. Uptake of CO2, is it, to hemoglobin is easier?
No, the uptake of oxygen is easier into the hemoglobins in curve A. Oh, sorry. In curve A, the uptake of oxygen into the hemoglobin is easier.
Okay. Can I modify the mark scheme? The first marking point, I think, is more accurate to say during aerobic respiration. During aerobic, because respiration, we've got two types, right? Aerobic and anaerobic. So for anaerobic, oxygen is not required. And in anaerobic, CO2 is not released for mammals. Because anaerobic respiration, mammals produce, lactic acid. Lactic acid. So you need to write aerobic. Because I know this question is not from Cambridge A level. So the mark scheme, you need to adjust a bit so that it is more aligned with the Cambridge A levels mark scheme to guarantee you'll get the marks. Okay. But I know that this workbook, the questions are really, really good for learning purpose. Okay, please continue. Thank you.
The next question is the figure below shows the oxygen dissociation curve of the hemoglobin of a mud-dwelling worm. Explain the physiological advantage of this curve. So if you look at this curve, at very low partial pressure of oxygen, the percentage saturation is already saturated. So for the first point you can talk about is that how it becomes saturated at very low partial pressure of oxygen. And because of that, it can act as an oxygen store. And another thing I can say is that because it acts as an oxygen store, it can take advantage of the low partial pressure of oxygen in its environment. So, for example, even if the level of oxygen is very, very low, it still has enough oxygen for it to function efficiently for all its cellular activities. And this is the answer key. So next, I'll be talking about fetal hemoglobin. So as you can see from this curve, the fetal hemoglobin is to the left. The fetal hemoglobin is made of 2 alpha chains and 2 gamma chains, unlike adult hemoglobin, where it's made from 2 alpha chains and 2 beta chains. So the fetal hemoglobin has higher affinity to oxygen than adult hemoglobin because the fetal hemoglobin has to pick up oxygen from the maternal hemoglobin through the placenta, where the partial pressure of oxygen is 2. So in this question, the figure below shows the oxygen dissociation curves of fetal and adult maternal human blood. Explain why the fetal oxygen dissociation curve lies to the left of the adult curve. So the answer is here actually. Because the fetal hemoglobin has higher affinity to oxygen than the adult hemoglobin. Because the fetal hemoglobin has to pick up oxygen from maternal hemoglobin through the placenta, where the partial pressure of oxygen is lower. This is the answer. And this question, shortly after birth, fetal hemoglobin is broken down and replaced by adult hemoglobin. Why is fetal hemoglobin unsuitable for use afterward? So as we know, the fetal hemoglobin has a way higher affinity for oxygen than the adult hemoglobin. So that is the first point. The other thing I can talk about is that in our atmosphere, the partial pressure of oxygen is actually like you don't need that much. The hemoglobin doesn't have to have such a high affinity for oxygen in our atmosphere compared to the placenta. So that's why it's broken down afterward. And this is the answer. Okay, so for the next question, let's look at the structure of the heart. So we have the right atrium, the left atrium, the right ventricle, the left ventricle, we have the septum, we have the aorta, the pulmonary artery here and here, the pulmonary veins here and here. You have the anterior vena cava, the posterior vena cava, and then you have your valves like the semilunar valves and the atrioventricular valves. So I'll talk about the cardiac cycle now. So the first step is atrial diastole. It is when the atrial muscles relax, and which causes the atrial pressure to be low. So now your pressure in your atria is lower than the pressure in your ventricles. So the AV valves close. The opening and closing of the AV valves is actually dependent on the pressure difference during the ventricular systole and during the pressure during the contraction. So for example, okay, I'll talk about that later. And then next is atrial systole. So as blood collects in your atria, the pressure increases because when the volume increases, the pressure increases. So your pressure in your atria is now higher than the pressure in your ventricles, and your AV valves open. So the blood enters the ventricles, and then the atrial muscles contract, which is atrial systole. And then next is ventricular systole. The blood enters the ventricles completely, and there's a delay of 0.1 second. The 0.1 second delay is to ensure that all the blood from the atria has completely flowed into your ventricles to prevent the overlapping during the atrial and ventricular system. So because of that, then your ventricular muscles contract and the pressure increases. So now the pressure in your ventricles is greater than in the vessels and in the atria. So the AV valves close and the semilunar valves open. And next is ventricular diastole, is when your ventricular muscles relax. So now your pressure in the vessels is greater than the pressure in the ventricles. Your semilunar valves closes and the pressure in the ventricles is still greater than the pressure in the atria. The AV valves close and the heart prepares for the next cardiac cycle. So for this question here, the diagram below shows the human heart in ventral view in a simplified form. Name chambers A, B, C, and D. So I'll open this one to the floor. A is here. Okay. You got it, left atrium. For B, right atrium, Shannon got it. D, left ventricle, Shumin. And C, right ventricle, Shannon got it. So for the next question, name vessels 1 to 4 and state whether each one is carrying blood to and from the heart. I'll open this one to the class now. So for 1, yep, Teng got it. For 2, 2 is here. Okay, Charmin got it. For 3, 3 is here. Joyce. Okay. And for 4, Teng. Okay, so for the first one, which is aorta, the blood is carried away from the heart. For the second one, which is pulmonary artery, is carried away from the heart. So basically, the arteries carry blood away from the heart to the respiring tissue. And then for 3, which is the vena cava, the blood is carried to the heart. And for 4, which is pulmonary vein, also the blood is carried to the heart. And the answer. Okay, so for the next question, state the functions of valve R and Q. So R and Q is the atrioventricular valves. So the first thing I can talk about is that we know how valves prevent the backflow of blood from like the ventricles back into the atria for atrioventricular valves during the ventricular systole. So that's the first point. And then for the next one, we can talk about how it opens to allow blood from the atria to the ventricles during atrial systole. And then for 2, state the functions of valves P. P is the semilunar valves. So the semilunar valves closes to prevent the backflow of blood from the arteries to the ventricles during ventricular diastole, and it also opens to allow blood into the aorta during ventricular systole. And what do S and T do? So S here is the tendon and T is the papillary muscle. So the papillary muscle it opens the valves and prevents the valves from inverting. during the contractions. And S is a tendon, it attaches the valve flap to the muscle.
And
And this is the answer here.
can you repeat your first sentence? It opens the what? Valve? It what? Papillary muscle.
It opens the valves and prevents the it from inverting during the contraction.
During contraction, it cannot open the valve, right?
Oh, sorry, it just opens the valve and prevents the valve from inverting.
Open the valve. I'll say that during ventricular contraction, papillary muscle will contract and pull the heartstring in order to hold the semilunar valve, in order to hold the AV valve so that the AV valve will not turn inside out. I think that is more makes sense than it opens the valve. When it contracts, it will pull the heartstring, right? So the heartstring will tighten. So that will actually also hold the AV valve so that it will not turn inside out. Yeah. Okay, thank you, Sarah.
Thank you, teacher. Okay, this one is the next subtopic. It is heart immunization. So for this one, you just have to know the location of the sinoatrial node, the atrioventricular node, and then the bundle of his containing the Purkinje tissues, which also covers the walls of the ventricles. So now I'll talk about initiation of heart contraction. So the SA node over here, it is a specialized patch of muscle near the opening of the vena cava and in the right atrial wall, also known as the pacemaker. It contracts and sends the excitation wave across the atrial wall, causing the right and left atrial muscle to contract immediately. And then after that, it sends it to the AV node. The atrioventricular node is a specialized muscle node in the septum of the atrioventricular wall. It receives the excitation wave from the sinoatrial node, and when the excitation wave is passed from the sinoatrial node to atria and atrioventricular node, it has a 0.1 second delay, and this is to ensure that all the blood has completely filled the ventricles first to prevent the atrial and ventricular system to overlap. And then next, it passes the excitation wave from the AV bundles to bundle of his and then to the Purkinje fibers. So these are specialized muscle fiber to conduct excitation wave. It sends the excitation wave down the septum and up to both the left and right ventricular walls, and then ventricular system.
So can I just add? There is a delay of 0.1 second when the excitation wave arrives at the AVN, right? Sarah?
Yeah, Ching.
Yeah, so when excitation wave arrives to AVN, there is a 0.1 second delay in the AVN before excitation continues to go to bundle of His, right? Yeah, okay, thank you.
Thank you, Ching. So for this question, the diagram below shows the conducting system of the heart in a simplified form. The heart muscle is said to be myogenic. What does this mean? So myogenic is like automatic or involuntary. So a more specific meaning is that it will contract and relax rhythmically of its own accord, so without the nervous system input. And the next one, name components A to D of the conducting system. I'll open this one to the floor. Um, no, Zisian, I'll show the image again. The sinoatrial node is here, and then the atrioventricular node is closer to the middle. Okay, Andrea got C and D. And Union got A and B. Okay, this is the answer. So with reference to the parts of the conducting system of the heart, explain why the right atrium contracts before the left atrium. So for this one, the first one is that the sinoatrial node generates impulses in the right atrium because that is where the sinoatrial node is located. And then only it passes it across to the left atrium. So basically when the sinoatrial node contracts, and then it sends the excitation wave to the right atrium first, and then the excitation wave is passed to the left atrium where it contracts within it. For 2, the ventricles contract after the atria. Impulses can only get to the ventricles via the atrioventricular node, which is located in the middle. So this delays the passage of the impulse, and it has a 0.1 second delay. So for this one, basically, in order for the excitation wave to be passed to the ventricles, the AV node has to contract, which causes the atria walls to contract first, and then only it is passed to the ventricles, and then the ventricles contract next. And why the ventricles contract from the apex upwards? So this one is because, as we saw the structure of the heart earlier, the Purkinje fibers leave the bundle of keys at the apex, and it branches to the ventricle walls upwards. So the only way it can contract is to contract upwards. And this is the answer. Okay, next subtopic is the tissue fluid. So if you can see here, this is the capillary bed. The blood is flowing from the arterial end of the capillary to the venous end of the capillary. So when the plasma is in the blood, it is called plasma. When it is in the tissue, it is called tissue fluid. And when it's in the lymph vessel, it's known as lymph. So tissue fluid is also known as interstitial fluid. It is constantly bathing the cells in the tissue. It comes from the blood plasma that has undergone ultrafiltration through the pores in the capillary walls. And you can find all soluble materials of blood plasma which have leaked out except for large proteins. For example, you can find glucose, amino acids, fatty acids, ions, water, and oxygen. And you can also find white blood cells in tissue fluid because they can squeeze out of the capillary, they can ooze through the walls of the capillary. So this question here, the diagram shows the capillary bed, and blood is flowing this way to the right. Some components of the blood flowing through the capillaries move out into the spaces between the cells. Name 2 blood components that move out of the capillaries. I'll open this one to the floor. The keyword is blood components. A1 is plasma and white blood cells. They are found in the tissue fluid, and they move out of the capillaries they are found in the blood plasma, and they can move out into the tissue. So for the next one, name 2 components of the blood that do not move out through the capillary walls. I'll open this one to the floor as well. Yes, Joyce. And Andrea. So the answers are large protein, plasma protein, red blood cells, all do not move out through the capillary walls. Next, name 4 transported substances that would move out of the blood into the tissues. So this one is more general. I also open this to the floor. Yes, Jinsen. You mean? I generally got it for glucose. So for this one, you can talk about oxygen, glucose, amino acids, lipids, some vitamins, hormones, all this moves out of the blood into the tissues. And these are the answers. Okay. So next, I'll talk about the effect of low partial pressure of oxygen in the environment on oxygen dissociation curve. So I'll be focusing on the llama. Llama is a mammal living at high altitude. So at higher altitudes, the air is thinner. So there's lower partial pressure of oxygen. The partial pressure of gas decreases with altitude, and the partial pressure of oxygen is lower at high altitude. So the llama has their hemoglobin must have very high affinity for oxygen in order to load oxygen from their external environment, which is low in partial pressure of oxygen. So this question, the graph below shows the hemoglobin oxygen dissociation curves of a human and a llama. Suggests why the llama hemoglobin oxygen dissociation curve lies to the left of the human curve. So the answer is right here. It's because the llama lives at a higher altitude, that is 1 point. And then the next one is that at higher altitude, the partial pressure of oxygen is lower. And because of that, their hemoglobin must have very high affinity for oxygen in order to load the oxygen from the external environment. So that is 3 points. That is the answer. Okay, so I'm actually finished, and now I'll pass it on to Rebecca. Oh, sorry, do you have any questions?
Wait. Capillaries are known as exchange vessels. Substances are exchanged between blood and tissue fluid as the blood flows through the capillaries. Figure 1.1 is an electron micrograph of a section through a capillary with two red blood cells. So this is a red blood cell, and this is another red blood cell. And these are what we have to find. And here you see this thin layer, it's the basement membrane. So name the cells label A and the structure labeled B. Does anyone want to try? That isn't the answer scheme. Something else? Yeah, you got that. Yeah. Okay, let's look at the answer scheme. So Andrea and Yun has gotten it. So here, you have to first describe the difference, which is the cardiac muscle in the left ventricle is thicker than that in the left atrium. So the atrium pumps blood at lower pressure or against less resistance to ventricle, which is a shorter distance with less force. Because the left ventricle, right, it pumps blood to or target organs in the systemic circulation. So it is located a large distance away. So the blood will experience larger resistance. So more force is needed to pump the blood to the targeted organs. Ventricle pumps blood to the body into systemic circulation, longer distance against greater resistance at higher pressure with more force. The reject ventricle walls withstand high pressure because blood is passed from the atrium to the ventricle, and it's at lower pressure, so the ventricles do not need to withstand high pressure. Instead, it needs the ventricle walls are thick to generate high pressure to pump blood over longer distances. So explain how the structures labeled on figure 5.1 ensure that blood flow in the correct direction. So the structures mentioned are atrioventricular valve, valve tendon, and papillary muscles. Sara has mentioned it, so I'll just go through it very quickly. So I'll talk about the valve tendon and papillary muscles first. So we know that from the initiation of heart muscles, the excitation waves will travel down the septum all the way to the apex of the heart, then outwards. So the papillary muscles will be affected first before ventricular systole. So the papillary muscles will contract and it will pull the heartstrings or the tendons, and then so it will not invert or turn inwards out. And the atrioventricular valves, as mentioned by Sara, its main purpose is to prevent backflow of blood, but you have to describe it in detail. You can write backflow of blood when you're describing like the veins, but in the heart you have to describe it in detail. So a valve opens to allow blood flow from atrium into ventricles. When pressure in atrium is greater than pressure in ventricles during atrial systole. Valve closes when ventricle contracts, when pressure in ventricle is greater than pressure in atrium during ventricular systole. So now I will attempt to explain both of them combined. Wait a minute. So here, the sense or sinoatrial node will send an excitation wave which will spread over the walls of both atria rapidly, and this will result in this can be represented by the P wave here. So an electrical impulse travels from the sinoatrial node to the walls of the atria, causing them to contract. But there are fibers around here which are non-conducting tissues, so it does not allow the excitation wave to travel straight from the sinoatrial node to the ventricles. So after a delay of 0.1 second, So when the excitation wave spreads over the walls rapidly, it results in the atricular systole where the left atrium will contract, or both atriums will contract. So this is atricular systole. Then, like I mentioned just now, the non-conducting tissues will not allow it to pass. So it will instead travel, the excitation wave will instead travel to atrioventricular node, and because of the fibers, it will result in a 0.1 second delay to allow blood to completely empty from atria to ventricles. After a 0.1 second delay, the AV nodes receive excitation waves from the sinoatrial node, which travels from the bundle branches, from the AV bundles to the His fibers, all the way to the Purkinje fibers and up, from the apex of the muscle up both sides of the ventricles, and it results in this QRS complex here. So ventricular systole happens right here, and it pushes the blood up into the vessels. So this is ventricular systole. So the question was asking for, during one cardiac cycle, blood is pumped from the heart into pulmonary and systemic circulation. Explain how the contraction of the 4 chambers of the heart are coordinated and controlled to enable blood to be pumped simultaneously into both the pulmonary and systemic circulation. So this is just talking about initiation of heart contraction. So I'll go through it. Sinoatrial node, or the sense, sends out waves of excitation, which the wave of excitation will cause the atria to contract almost simultaneously, and there's a fibrous ring of non-conducting tissues which prevent impulses from reaching the ventricles directly, and prevents the ventricles from contracting at the same time as the atria. So atrial systole and ventricular systole do not occur at the same time. Atrioventricular nodes delays impulses by 0.1 second and prevents ventricles from contracting at the same time and allows atria to empty and ventricles to fill. AVN sends out waves of excitation impulses to Purkinje tissues, bundle of His, and in septum. And this causes ventricles to contract at the same time simultaneously. So identify tissues present in layer A and B. Does anyone want to try this? Has gotten it. So they're talking about tissues, so you can't write cells. And you can't talk about layers as well because you're asking for tissues. So A is the endothelium, or simple squamous and epithelium, or pavement epithelium. Is found is the tunica intima, but you can't write that because they're asking for tissues. And B is the smooth muscles and elastic tissues, which are found in tunica media, but you can write that. Describe how the structure of an artery is suited for its function. So I'll go through what the functions of an artery are. Function of arteries are to carry blood away from heart and tissues at high pressure, and 120 mmHg is the pressure in aorta. Tunica intima is the inner coat, which is formed from squamous endothelial tissues, and it has a smooth surface to reduce friction of blood flow. Tunica media is the middle coat, which is made out of elastic tissue and smooth muscles. Elastic tissue allows for elastic recoil, where it allows for stretching when pulses of blood flow in at high pressure away from the heart when ventricles contract. It reduces pressure on atrial walls and stores potential energy. It recoils when pulses of blood flow away at lower pressure when ventricles contract. Pushes blood forward and maintains diastolic blood pressure. So it maintains blood pressure between pulses. That's why you have diastolic blood pressure. Smooth muscles is to regulate lumen diameter by vasoconstriction and vasodilation. Tunica externa is the outer coat, which is formed from collagen tissues and provides structural support. Small lumen to increase resistance of blood flows to increase the pressure of blood. So how is the structure of artery suited for its function? It contains elastic tissue to absorb pulses or energy of pulse, withstand high pressure. It contains much smooth muscles to withstand high blood pressure and allow vasoconstriction in small arteries. Narrow lumen to maintain high pressure. Smooth non-stickable endothelium so blood can flow through easily with little friction. How does the structure of a vein differ from that of an artery? Suggest why veins are differ in structure to arteries. So you guys want to try? The structure between arteries and vein. So these two questions are very similar, so I combined them. Blood is at much lower pressure in the vein, so less smooth muscles and elastic tissues are needed, as there is no pulse, so no need for elastic tissue. Valves prevent backflow of blood due to gravity effect, contraction of atria. Wide lumen allows lower pressure. As you can see, it has a larger lumen, thinner elastic fiber and smooth muscle, and has thicker, it has collagen fibers here, which are around the same. So now question 3. Hemoglobinopathies are inherited conditions linked to the structure and function of hemoglobin. Sickle cell anemia is one of these conditions in which the transport and delivery of oxygen to tissue is less than normal. An investigation was carried out to discover the effect of sickle cell anemia on the ability of blood to carry oxygen. Blood samples were taken from 2 people, person L without sickle cell anemia, person M with sickle cell anemia. The percentage saturation of hemoglobin with oxygen was determined over a range of partial pressures of oxygen. Figure 3.1 shows oxygen hemoglobin dissociation curve for 2 blood samples. So this is for person L, who does not have sickle cell anemia, and this is for person M, who has sickle cell anemia. So P50 is the partial pressure of oxygen at which hemoglobin is 50% saturated with oxygen. It is taken as a measurement of the affinity of hemoglobin for oxygen. State the P50 for the 2 blood samples L and M. Does anyone want to try? No?
3.6 and 4.5 kilopascal.
Yeah, teacher, you got it. Couldn't move it around in the pictures. So describe with reference to figure 3.1, describe how the dissociation curve for person M differs from the dissociation curve for person L. So for this, you have to basically describe what you see in the graph, in the picture. So let's look at the marking scheme and then I'll go back and forth to show you. To the right, the dissociation curve for person M is more towards the right, so it has lower affinity for oxygen. At lower or higher or lower partial pressures, there's a smaller difference in percentage saturation. As you can see at the start and the end, there's a small difference between the two curves. So that's what they mean. And that is comparative data code. So you must refer to L and M, allow example from number 1. So you can say that percentage saturation of hemoglobin with oxygen at 50% is when partial pressure of oxygen is at 3.6 kilopascal and 4.5 kilopascal, or you can talk about when percentage saturation of hemoglobin with oxygen is at 95%, then the partial pressure of oxygen is at 10 kilopascal for curve L and 12 kilopascal for curve M.
Is this a CIE passive question?
This one? Yeah, this is very long ago, 2012.
So that means the students need to actually remember the loading tension and unloading tension, because that's what you're
So I'm not
mentioning.
sure because this one is the answer I gave on my own because that's all they gave from comparative data code.
Ah, I see. So any value will do lah. Okay.
Yeah, any
Thank
value
you.
would do. Explain the, oh my God, sickle cell anemia. So a person M with sickle cell anemia has a curve which is more towards the right, as shown as the green one. Hemoglobin of person M has a lower affinity for oxygen. So you can think of L, as teacher mentioned before, as oxygen loving. So left-hand side is oxygen loving, right-hand side do not love oxygen. So HB, which is hemoglobin, is less readily associates with oxygen. So it is less likely to associate with oxygen, but it's more likely to dissociate from oxygen. At lower partial pressures, it is more difficult for oxygen to bind to hemoglobin. So less loading of oxygen into hemoglobin to form oxyhemoglobin, and more unloading of oxygen from hemoglobin to supply more tissues with oxygen for aerobic respiration to produce ATP. Explain the advantage of the position of dissociation curve for people with sickle cell anemia. So for this, you want to talk about the lower affinity, so you can release more, so more hemoglobin can dissociate to form oxygen. So this is the answer. At partial pressures in the tissues where oxygen is unloaded from hemoglobin, hemoglobin is less saturated. So you're supposed to talk about the saturation of hemoglobin. Because hemoglobin dissociates more readily, the idea of unloading oxygen more readily, even if hemoglobin is not mentioned, it is allowed to compensate for fewer or less red blood cells. C, the partial pressure of oxygen in the lungs at sea levels about 13.5 kilopascal at an altitude of 3,000